Okay. Lecture caption. Lights, camera, action. Okay, so immortalize the cells, make them divide indefinitely, also could have inherent problems, could have aneuploidy, incorrect number of chromosomes. What else? You said can develop a mass or a tumor because they lose those cell checkpoints and cell cycle stops, so they just keep dividing and dividing. So ultimately can lead to either immortal cells that are kind of don't undergo the normal aging process, or it can lead to cancerous situation, masses, tumors. And a lot of this goes hand in hand with, yeah, you're gonna have some chromosomal or genetic problems over time. Okay, what are the three major differences between eukaryotic and prokaryotic DNA and how they replicate their DNA? What are the major differences? At least three. Yes?
The size, the location, and the complexity.
Okay, that's in general, yep. So the size of the DNA, the location of where the DNA is kept, and the complexity of the DNA. Okay, but what are the details? So that's very general. That's a good generalization of those three categories. That's perfect. But what are the details? Can you draw these out? Yes, you.
Eukaryotic is linear and then prokaryotic is circular.
Okay. Eukaryotic, right? It's linear and it's in separate chromosomes, right? So it's linear. You have all this double-stranded DNA. These are arranged in the chromosomes. And then in the prokaryotic situation, it's circular. It's double-stranded, but it's circular, right? So it's smaller. There's less genetic information. It's in a circle. It's not linear. All these are double-stranded. And then what you said that location in a cell, right? There's differences in location of where they're kept. Where's a prokaryotic DNA? Where would you find that?
Just floating around.
Just float around. It kind of condenses in an area we call in bacteria like the nucleoid, but it's not a nucleus per se, right? So this is inside of the bacterial cell. That's it. It's just contained in there. It kind of clusters in a certain area. Where is the actual double-stranded DNA in a eukaryotic cell? Plants, animals. In a membrane-bound nucleus. So it's actually contained in a nucleus that's inside of a bigger cell. This is horrible. But this shows like bacteria, very small compared to eukaryotic cells. There's rough ER, right? There's the Golgi, there's the mitochondria, there's all that stuff going on, but the nucleus is what contains the linear double-stranded DNA, okay? So very different in how they parcel out things within their cell parts and where the DNA is, what it looks like. Now, what are the major differences between how they replicate the DNA? Hopefully you all said there's some real major differences between prokaryotic and eukaryotic. What are? This is recap because we talked about this last time. Eukaryotic?
Membrane-bound nucleus.
No, that's more locations, that's DNA. Well, where it's occurring, yes. Okay, so it's occurring, yeah, in the nucleus here. Not in the nucleus here because there isn't a nuclear envelope in the nucleus there. But the actual intricacies of how DNA is replicated or copied.
Isn't the prokaryotic like super short but also really fast?
It's very fast. There's less of it. It's very fast.
Is it one replication bubble?
Yes. Yes.
And the eukaryotic has like as many as it wants.
Exactly. So one replication, origin of replication. That means on this circular DNA, the enzymes pop on and open it up at only one location. And then it's very processive. They all add on there and they just zip all the way around. So they basically, this replication bubble gets bigger and bigger and bigger until it's ultimately, this results in two double-stranded U-circular sets of DNA. One origin of replication, one replication bubble to make duplicated circular DNA. Okay, so it's real simple. It's like a typewriter, just opens and brrr. Now, hopefully you know by now, we went through the replication lecture series, eukaryotic, and I really emphasize this one the most. It's the more complex of the two. This one has multiple points of replication, so multiple origins of replication along any given linear DNA, stretch of DNA, or any piece of chromosome. So we'll unwind it, we'll say it's like that, double-stranded. All along here, there are multiple origins of replication. That means where the topoisomerase and the helicase, right, grab on, kind of go to the end, open it up, pop it open, and the DNA polymerase, the primase, and all those enzymes can come on, open it up, and start replicating. This is happening in multiple locations along. And these get bigger, right, and bigger, until this ultimately merges, and you get two new double-stranded sets of that exact linear segment of DNA, or that chromosome. right? So multiple origins of replication, multiple replication bubbles, and remember there's some components that are different. The way the origin opens and allows those enzymes in, and because DNA goes processively from the 5' to the 3' end in being made, so first there's a 5', right, and then it's added onto the 3' end. Because of that polarity to DNA replication, remember in eukaryotes, it's very weird. There are leading strands that go continuously till they hit the end, and then they keep copying. And then there are lagging strands that are made behind that leading strand, right? Because as it opens and it exposes more template, enzymes can now sit on and copy. Then it opens up more and more to sit on and copy. That doesn't happen in prokaryotic. So eukaryotic is unique in that way. What are those short? We've got the leading and the lagging strands. You will not see that in a bacterial DNA replication process, right? So you've got leading and lagging. What do we call those little lagging strands or segments of DNA? Okazaki fragments. And remember, this leads to, you end up with this little 3' overhang, right? Where you can't copy the ends of the DNA. So it causes shortening of the chromosomes at the end if it's not for this enzyme right here, telomerase. We talked all about that, right? The implications of that, why it's important. It's involved in aging and allowing a cell to just keep on dividing indefinitely. Okay, so a lot of major differences between the two. Okay, so now I'm going to go back. That was the application questions from DNA replication. Now I'm going back to where we were, and that's transcription. Okay. Transcription and RNA processing. Okay. So I think we kind of left off right there. Okay. So, types of RNA. So remember, do you guys remember the major differences between DNA and RNA? And if I showed you a visual, if I drew them, let me back up. The sugar phosphate, ribose, phosphate, ribose. And then these are just says base, but if I put these purines and pyrimidines and attach them to where the nitrogenous base would go, would you be able to recognize DNA from RNA?
Yeah.
Okay, good. I would like you to be able to do that. You should be able to know the differences between DNA and RNA. And in terms of the bases, if you ever see a nitrogenous base uracil, right? And that's one of the 4, there's a U. You know right away which nucleic acid are you looking at? DNA or RNA? Those are nucleic acids. RNA, you know that, right? So you know that right away. So remember the differences between RNA and DNA structurally, and then also functionally. How are they structurally different? And remember, DNA is deoxyribonucleic acid. So a deoxy, we would just, boom, get rid of that hydroxyl, we just turn it into DNA. As long as we don't have uracil, that's pretty much DNA, right? Sugar phosphate backbone with the deoxyribose, the 5-carbon sugar that's known as ribose. The function of DNA, someone tell me what's the function of DNA? Store genetic material. It is the genetic fingerprint. It is the genetic blueprints. If we're like building a house and we have to construct everything, the blueprints for how to make everything, where everything's gonna go, what it's gonna do, that's the DNA. It is the instructions for every cell inside of your body. It's the instructions for you. It's for the instructions for the organism, the blueprint, the sequence that leads to proteins. What is the function of RNA? So DNA is basically the instructions for everything. What about RNA?
Protein.
What is to make protein?
Yeah.
Okay. Mostly the function of RNA, the various different types of RNA, yes, like you said, Ian, it's mainly to make all the proteins. So make all the proteins in your cells, in your body, basically allow all those things to be created, right? To lead to a phenotype. So this is like genotype. This is your genetic instructions, RNA. This is your phenotype. Wow, it's inaccurate at the bottom of the board. It's not where my finger is. Phenotype, what is phenotype? Genotype is clear. Genotype is your genetic alleles at particular sequences, right? Or your genome. What is the sequence of your DNA? That's your genotype. What are the different alleles at all those different genes and locations? This is so messy, I'm sorry. But RNA, I'm speaking while I'm writing. RNA leading to all of its various diverse roles and the different types of RNA, because there are many types of RNA. The whole point is to help express proteins, and those proteins make up your phenotype. So what is your phenotype?
What display?
What you display, what that cell displays, what it looks like, how it functions, what are all those working parts? Everything from your hair, your skin, your eyes, your body, all those different things, that's part of your phenotype. And they're coded for by your genes. In combination with environmental factors, of course, right? What you're exposed to. Your genetics, instructions, those have a complex interplay with the environment. But for most part, genotype is the sequence of your DNA, the instructions, the alleles, that what leads to these proteins. And RNA, the functions are to allow those proteins to be expressed, to give you the physical characteristics that you have, even down to the cellular level. So starting at the cell level, going up to tissues, organs, organ systems, and the whole individual. Okay, so they have very different functions. This is just a recap to remind you their structure and their functions are very different. Types of RNA, one of the main ones that we're going to spend a lot of time with in this lecture is transcription, making DNA, copying DNA into mRNA. Messenger RNA, hence the M, right? So messenger RNA, this is an example of, here is basically the production at the end. We're going to go through how this is made, but we have an mRNA right here, an mRNA template, or a strand. Okay, so it's a nucleic, it's basically a nucleic acid sequence, single-stranded, and it's got sites, it's got segments into it. Every single one is different. It's got segments into it that are known as exons and introns. The exons are in red, the introns are in blue, okay, represented that way. And there is a 5' and a 3' in it. It's a nucleic acid, right? It's like DNA, it's just RNA. It has it's single-stranded here. So what ends up happening is it gets spliced. So ultimately, some of these segments, specifically the introns, get clipped out. It's supposed to be scissors. And they actually loop up with enzymes. They loop up and they get cut out, and then the RNA, the rest of the sequences, the exons, get ligated or glued back together with this whole enzyme complex machinery that I'm going to show you at the end. We call this RNA splicing. So this is RNA processing and splicing. And I have a whole section at the end that talks about this and splicing. That's what we call it. So it's basically fruit ninjaing the RNA, just cutting out the pieces and going, okay, this now is the coding sequence of the messenger RNA. This little particular, and here in this case, it's only 147 nucleotides long. It's pretty short in this case, but this is what's going to be the specific instructions for the amino acids that are going to be used later to build the protein polypeptide from this, okay? So we also do, your cells do some other things. There's some fancy enzymes, and they basically create a cap. They put a little guanine cap on the end. They cap the end, and they add a polyadenylated, so adenosine out of the end, they add a polyadenylated tail. So these mRNA sequences, they get capped, tails, spliced, and then ultimately you have this final mRNA. This is your final mRNA sequence that leaves the nucleus and goes out, and this is what gets turned into, translated into protein. A lot of fancy things are happening, okay? And we don't have all this fancy RNA processing in bacterial cells or in prokaryotic cells. This is happening in eukaryotic cells. RNA processing, RNA splicing, getting out the introns, keeping the exons shown in red, so you have a final transcript. You cap it and tail it, send it out into the cytoplasm, where it either gets picked up by a free ribosome in the cytoplasm or a ribosome bound to the rough ER and turns this into the designated protein that it's supposed to be. Okay, so that's one very important type of RNA, mRNA. But how does all this happen? And then how do you get RNA to go to protein? You need a bunch of other RNAs. So there's tons of different types of RNAs. And actually some of these RNAs have RNA enzyme-like activity. They're not just RNA, but they catalyze this process. They catalyze being able to turn a messenger RNA that we just talked about, mRNA, into polypeptides that are being made. In this case, the amino acids are represented here by AA1, AA2. This polypeptide being built right here, the way that your cell can build together, this is a protein being made, okay, or a polypeptide being made before it's folded and complex and everything. All these other types of RNAs are aiding in this process of gene expression or translation, okay? So transcription, we're going to talk about now in a second, uses this mRNA, makes this mRNA. and then translation turns these instructions into building a protein. And in this process, there are tRNAs, and they actually are loopy like this. They kind of look like a T. They make like a T-like structure. So that's how I remember. Transfer RNA, they actually look sort of like a T. They have these loops, hairpin loops, that make them look like a cross or a T. They're the ones that bind onto and carry amino acids, specific amino acids according to their little anti-codon sequence right here. Three base little codon. We're gonna talk about these more later. mRNA is red in sets of 3, so those are representing the codons. And then also in the ribosome, there's a large subunit of the ribosome, and there's a small subunit. This is the ribosome that we've talked about already, right? In the rough ER, where there's ribosomes, and you can see them, they're like the factory workers of the assembly line of the factory. If the cell was a factory, these are the factory workers. These are the ones that are reading the mRNA codons or sequence, and they're allowing the tRNAs to bind on at the appropriate location, carrying an amino acid to allow the building of the protein. These are made of rRNA and protein together. So ribosomal RNA and protein in these big complexes. And so all of these things put together, this is just a few examples. There are other types of RNA I'll mention in a second. But all of these are working together to allow translation into protein. So we've got the mRNA, we've got the tRNA, we've got the rRNA. You should know what those are. Messenger RNA, ribosomal RNA, transfer RNA, right? The M, the T, and the R, the different types. Those are the 3 major types. There's some other types that also come into play in cell function and have a lot of different roles. I put a chart, so this chart kind of goes over a lot of these. We reviewed a lot of types of RNA earlier, and I kind of mentioned, well, we have mRNA and tRNA, and I didn't expect you to know a lot about them, just the difference between RNA and DNA. Well, now I want you to know them. You need to know these because these are involved in gene expression, transcription, translation, and getting proteins made inside your cell, going from genotype to phenotype, okay? So we have mRNA, we have tRNA, we have ribosomal RNA. We also have some other different types. snRNA, or small nuclear RNA. These are localized in different places in the nucleus, and they play structural and catalytic roles in spliceosomes. So spliceosomes are like the fruit ninjas that fruit ninja up the mRNA. So remember I talked about splicing a second ago, how RNA is made but then it's processed? snRNA, or small nuclear RNA, are part of these spliceosomes, these big protein enzyme complexes, RNA protein enzyme complexes that help to basically fruit ninja up and remove the introns and splice the exons back together so you have a final continuous sequence of an mRNA. Okay. Any questions on those so far? What they do? What does mRNA look like? RNA processing, different types. We have a few different other ones. We've got small nucleolar. So remember in the nucleus there's a nucleolus as well. There's a concentrated area in the nucleus where I told you a long time ago you have little pieces of RNA and segments of chromosomes that kind of cluster right there. This SNO RNA, or small nucleolar RNA, this helps in pre-processing pre-RNA transcripts for ribosome subunit formation in the nucleus. So pre-rRNA, that's ribosomal RNA. This is helping in basically transcribing those rRNA transcripts so they can get ready for making ribosome subunits. So the assembly of ribosomal subunits, that large and small subunit of ribosomes, starts to get put together and created and then exported. So this is involved in that at the nucleoli inside of nucleus. The SNO RNAs help with getting those rRNA transcripts ready. And then there's also some other ones that do a whole bunch of interesting things. And when these go out into the cytoplasm, so they get made and then they go out, they are involved in all kinds of cool functions in regulating gene expression. So regulation of gene expression, that means sometimes they shut it down, they turn it off, they inhibit the translational machinery and say, nope, don't transcribe this mRNA, don't turn it into protein, don't translate it into protein. And then other times they encourage gene expression. So there's small interfering or siRNA, microRNA, and even some known as peewee RNA. These are little tiny nucleotide sequences. All of these are short, little tiny nucleotide sequences that are specific to certain genes. They're specific to certain mRNAs. So if they bind onto those mRNAs, they can either facilitate its translation into protein, or they can bind on and block it from being translated into protein. And actually in science and biology, we use a lot of, we capitalized on these inside of cells Cells have their own way to either block or activate translation of certain genes, and we're gonna talk more about this when we get to translation, this next set of material. But we, in science and biotechnology, we've capitalized on using some of these siRNA and microRNA. We can engineer them and make them. We can just create these short little nucleotide sequences. If we wanna, say, block a certain gene from being translated, so we can actually engineer these because we know the sequence. As long as we know the sequence of the DNA, we know the sequence of the mRNA, we can construct siRNA or microRNA to go inside of the cells and block certain genes from being turned into proteins. And so we have really harnessed this and capitalized on this. We can knock genes out, we can add genes in, we can block gene expression either in cells or in whole organisms. We've done this even in my lab. We put in these short little interfering microRNAs and blocked different pathways, different proteins that were involved in different aspects of development. We can shut them off and we see what happens. So a lot of times you can assess function, and you guys should know this by now in this class especially, right? If you pull something out or you knock something down or you inhibit it, you can really see what that component does normally, right? Because then you can see what goes to crap. When you knock it out, ooh, all these things cascade and you have all these problems. So this is how biologists and scientists have figured out all the different things that many proteins and many things do is by shutting them down or knocking them out. And we actually call them KOs, like knockouts, right? Because I'm gonna use a microRNA or a silencing RNA or a few, because I'm gonna knock down gene expression, and for me, I'm interested in stem cells and development in embryos, and sometimes there's a key protein that does something that's really, really important. If I can just silence it, shut off the translation of that mRNA into protein, I can see what that protein does normally, right? That's one way to really get at function, is to knock it out. Okay, so these are two other categories of really important teeny little nucleotide RNAs that have big impacts in terms of gene expression regulation inside of a cell, and we use them as scientists all the time. Okay, so what are genes? What are genes? What's a gene? So now we're going to talk a little bit more about what is a gene and revisit this idea several times as we go through these lectures. What is a gene? What is a gene? Simply, like if you had to tell your grandma, a gene is anybody know?
A segment of DNA that codes for something.
A segment of DNA, okay, so a segment of DNA or a stretch of DNA, a sequence, right, of DNA that, what?
Codes for something.
Codes for something, okay. So a sequence or stretch of that sequence, right? A sequence, a stretch, or a particular area of the genome of DNA that codes for, that's a good way to say it. And I'm gonna say something because I like something. It could be a protein, and as we'll see now, it could be an enzyme. We've already talked about enzyme. It could be an RNA, one of the different types of RNA. So really, it's coding for typically proteins, but also some of these other things. And many enzymes are actually, most enzymes are actually proteins, right? So it's a stretch or sequence, a segment of DNA that specifically codes for one of those building blocks, one of those proteins, something inside of your cells. There's all kinds of software in bioinformatics. This is done by GeneScan to basically predict genes in sequences and sections of so going all across a particular location on a certain chromosome. It actually is looking down here. There's an initial exon, okay, and then there's a terminal exon, which is down here. And this is all the whole region here. And what you see is there's these little blocks, right? Segments of nucleotide stretches, these stretches of DNA. But is it continuous? All these little exons, are they all continuous? Not really, there's gaps, right? There's a huge gap here in the genome along this chromosome, along this DNA. What's here? What's there? What's there? There's all these gaps, but there are blocks of continuous or contiguous sequences of these bases, of these stretches of DNA. This is one gene, so what the point of this is to show a lot of this in between, it's not the exons. There's a bunch of introns and other stuff that has to be, ultimately, that's gonna not be expressed into mRNA. Not gonna end up in the final mRNA and the final protein. And we can do sequence alignments and see how genes are similar, how they're different, how these line up. But a lot of your DNA, a lot of your chromosomes and your genome has a lot of non-coding, so non-gene areas that don't code for anything. Matter of fact, the vast majority of your genome doesn't code for anything. It's kind of crazy because there's tons of sequences, sections, where they're not part of any gene, they're in between genes, like they're upstream here, or they're downstream here, and there's no actually genetic sequence or gene right there. And a lot of this junk has to be trimmed out. Although what we used to call junk or non-coding DNA, we now know has some important regulatory regions that controls the expression of adjacent areas. So even if it's junk DNA, it doesn't mean that it does absolutely nothing. There's some relevance and some things in regulatory mechanisms that are still involved. But genes are pretty complex. It's not like you just have one continuous gene with all the bases right here, boop, boop, and there's the gene, and then from that comes the mRNA, and there's the mRNA. There's a lot of splicing and pieces that are kind of disconnected. They're not all in a direct alignment. Okay, so how can we make mRNA? That's the whole point of this particular lecture series. How can we go from DNA and replicating the DNA, so now the cell divides, it's happy, and now it's in its interphase and it's growing. Say it's in growth 1, just making proteins, resting and digesting, doing its thing. The cells need to express proteins, right? They need enzymes, they need all the metabolic pathways that can be carried out, they need to respire, take in oxygen, let out CO2, do all of that stuff. So they need to constantly be taking DNA into mRNA and mRNA into protein, the central dogma. So that's always happening. So the 3 stages of how they go from DNA, so remember we're now at this part, DNA to mRNA. That's where we're at in the central dogma, or the flow of genetic information, okay? The 3 stages of this, copying that DNA into a message, to an actual message, is we have initiation, elongation, and termination. That's easy, right? IET. Initiation, so there's initiation machinery and enzymes that pop on and start the process. Elongation, those enzymes continue along and elongate that whole stretch of mRNA that's being made. Termination, they stop. They pop off, they're done. They're at the end of the genetic sequence, they've made their whole sequence of mRNA, boom, they terminate. So those are the 3 stages of transcription. I'm gonna show you videos and I'm gonna go through these steps, exactly what's happening in all these steps of transcription. Now you should really know in your mind though, what's DNA replication, what's transcription, and what's translation. You should know what those processes are, what they lead to in a simple document. Okay, molecular components. Again, we have enzymes that you have to know, right? We talked previously about helicase, topoisomerase, DNA pol 3 and 1, right? Primase, ligase, the sliding clamp proteins, the single-stranded binding proteins. We talked about all those things. Now we have a different set of enzymes, and their specialty is converting DNA into mRNA, okay? So, RNA synthesis is catalyzed by RNA polymerase. Remember, what does a polymerase do? What does a polymerase do? Polymerase creates polymers. In this case, the polymer is RNA. I'm just gonna move this, I feel like I'm gonna trip on this chair. Creates polymer of RNA, okay? So it creates nucleic acid, ribonucleic acid. It is making the RNA message. And basically what this does, this RNA pulls the DNA strands apart and then hooks together the RNA nucleotides. The free-floating RNA nucleotides that are available, grabs them based on the sequence of DNA. Okay, so RNA polymerase is the enzyme that does the majority of making the mRNA message. Same base pairing rules as DNA. So base pairing being which purines or pyrimidines, the nitrogenous bases, pair with which ones. Remember, they've got a specific way that they pair, they make those hydrogen bonds with each other. A goes with T, whoops, C goes with G, yes? But in RNA, okay, this is DNA pairing rules. I'll put this over here because you can't really see it. But in RNA, what are the pairing rules? Base pairing, how they complementary bind, make those little hydrogen bonds with each other. And by the way, we have to do that here because we're basically building RNA from the DNA sequence, right? So they complementary bind up, they base pair. What is it in RNA? So that was for DNA. What about RNA? Does C go with G still?
Yeah.
Yes.
A, U.
There you go. A goes with U. Remember, thymine is swapped for uracil in RNA. So the base pairing rules here are the same, except we don't have a thymine that's involved in RNA. It uses uracil, so it's A with U and C with G. Yes? Okay, so that's the exception. And then there's a promoter sequence and there's a terminator sequence. So those sequences on the DNA in the gene, right, at the level of the, right before the gene and at the end of the gene, that right there are sequences that let these enzymes know where to bind on and start, where that gene begins and where that gene ends. Okay? So where to come on, those promoters come on, they allow the transcription into mRNA, and then where to terminate, where should we stop? So, and it's typically along an actual segment of DNA that's a gene. Okay, so transcribing along a particular sequence of DNA, that is a gene. So a stretch of DNA that's transcribed, what we'll call right now is a transcriptional, we call this a transcriptional unit, or transcription unit. So a lot of times it's the DNA, it comprises a gene, but it might also be a little more than that. And ultimately it's this whole sequence, it's this whole area. But that whole area, or that whole stretch, doesn't mean that that's a continuous sequence of the gene. Remember, because I showed you, it's discontinuous in many sections. There's exons and then there's introns in there that have to be cut out. So we call this whole thing, because it's gonna be spliced later. It's not in its final form, like that whole segment is not the entire gene, it's the gene within that area, so we call it a transcriptional unit. This is the unit that has to be transcribed into mRNA. And then that's gonna be polished, refined, and spliced, and capped, and tailed later. Okay, so RNA polymerase binds and then causes the initiation of transcription. So we have promoters. These promoters are promoter sequences, they're DNA sequences that allow initiation of RNA synthesis. So that shows where the synthesis of that mRNA needs to begin. So upstream, right in front of that gene, or that transcriptional unit. Transcription factors, these are proteins that actually allow the binding of RNA polymerase and the beginning of transcription, or the initiation of transcription. So the promoter is the sequence on the DNA that's right upstream of that transcriptional unit, saying, hey, okay, this is where we start, this is where we need to transcribe and turn this gene or this transcriptional unit into a transcript. The transcription factors are the proteins that actually bind on and help RNA pol to sit down and make the mRNA. They go, okay, here's where we sit, and the transcriptional factors bind onto that sequence, the promoter sequence, and then that allows RNA pol to clamp down, grab on, and now zip along it and do the elongation portion of transcription, where it's grabbing nucleotides and building the RNA sequence, building the mRNA. And then we have this whole transcriptional initiation complex. Transcription initiation complex, this is that entire complex of these transcription factors of RNA polymerase II, that's the specific one involved, all bound to this promoter sequence. So everything's sitting on this little segment of DNA. So here's the transcriptional unit, maybe the gene itself right